We're going to get started. Welcome everyone who's online joining us via Zoom, and then welcome to everyone who's here. Thank you so much for coming today. It's with great pleasure that I introduce Dr. Arial Shogren, who is visiting us from University of Alabama in Tuscaloosa, where she just started as a faculty member in the fall of this last year. So she's a brand new faculty member there. And I realized when I was thinking about what I was going to say about Arial is that in thinking about it, she has done every possible alumni thing that she could do at the Carey Institute. So she started as an undergraduate sieving soils with Lynn Christensen, who, as many people know, is at Vassar. Then she became an REU student at the Carey. Then she did her honors thesis at the Carey. So she did her REU with Shannon and Gary, and then she did her honors thesis with me. And then I hired her as a research support specialist. So she was a technician for a year or for a couple months. Then she went on to graduate school at University of Notre Dame, but we still couldn't keep her away. She came and ran a bunch of experiments in the artificial stream facility, and I was honored to be on her committee, her doctoral committee. Then she went to and did her postdoc. She was funded by an NSF postdoctoral fellowship, and she was at Michigan State University working with Jay Zarnetsky. And now she's at University of Alabama, and she's done research on a lot of really cool, aquatic. I asked her kind of how she defines herself, and she couldn't really give a good answer because she's worked on a lot of things that are really good at understanding the influence of flow on a number of different aspects of aquatic ecosystems from eDNA, and she's going to talk about her work in Arctic systems today. So excited to have you back, Ariel. Thanks so much for coming.
All right, hopefully you all can hear me. Yeah, the mic's on. Well, thank you so much for that kind introduction. I am so excited to be here. Emma didn't mention, I gave my very first scientific talk here at the Carey, so it's really wild to be back, especially at this stage of my career. So I'm going to talk about a little bit today about mostly work that I have either ongoing or that I completed during my postdoc. But just to give you a sense of how I think about the world, so I view water as my study organism. I'm really excited about how flow moves material from the landscape down river channels. And so the work that I do, the core of my research theme is essentially trying to understand how ecology and hydrology interact to tell us something about what's happening on a landscape or what's happening in an ecosystem. So this is my happy place, like between ecology and hydrology. So I use a lot of tools like empirical work. I do a lot of experimental work, as well as modeling to understand how flow changes material transport dynamics. So I do want to mention that I've done a lot, as Emma mentioned, kind of a jack of all trades in some ways, where during my graduate work, I worked a lot on environmental DNA and genetic material, as well as particulates. I very intentionally moved to a postdoc where I could work on dissolved solutes. And now at Alabama, my group works on questions about how climate change and dynamic flow regimes change transport patterns, as well as some invasive species work as well with invasive hogs that are a huge problem in Alabama, fun fact. So, but for the sake of time, I'm going to talk about two NSF-funded projects today that focus specifically on the Arctic. So I'll focus mostly on carbon and nutrients, but keep in mind that the things that I talk about today could be applied to a variety of systems, including those in New York and yeah, across the globe. So I want to take you first to this landscape that most people would think about when they think about Arctic systems, that it's cold and vast and frozen. And that's true for about the majority of the year, for like eight months. But for about 4 to 5 months of the year, the Arctic greens up and ends up looking a lot more like this. So this is sort of when the Arctic comes alive. And while the surface is brought to life, I just want to highlight that Arctic systems are uniquely constrained by kind of what's under the hood or what's underneath. So this cutout shows a soil cutout of what we would think about in terms of a typical Arctic soil profile. So you'll notice here that permafrost is essentially defined as frozen soil that's been frozen for consecutively 2 years. When we talk about permafrost degradation, what we're actually talking about is this active layer right here. This is like that nice brown organic layer. And what permafrost degradation means is that that active layer is essentially getting thicker and thicker, accessing deeper layers of formerly permafrost. It's no longer permafrost because it's not frozen. The active layer is dynamic during the season, so it thickens during the course of the season. And yeah. But because it's this amazing store of old frozen organic material, it's sort of this sleeping giant of carbon and nutrients that's previously frozen. And as the active layer thickens, we're now accessing deeper layers of this old material that then can become active in local and global biogeochemical cycles. So it's this really important sleeping giant of material. And interestingly enough, permafrost underlies about 25% of the northern hemisphere. So it's a massive store of material that it's estimated that carbon stores in permafrost soils are about twice as much carbon as what's in the atmosphere currently. But it's no secret, I don't have to probably tell you that the Arctic is rapidly changing as a result of many co-occurring dynamics of change. So the first thing, right, is that active layer is deepening. Along with that, there's more discrete influence of disturbance, such as thermokarst, which is essentially thermal erosion processes that where the permafrost collapses very quickly. There's also intensified hydrology, and what I mean by that is that there's extended flow seasons, meaning that the spring is happening earlier and the refreeze is happening later, as well as more sporadic storm events that have the potential to move material. In addition to this, there's also shifts in vegetation structure from very, very small little plants to more shrubby, taller plant structure. So all of these things, essentially, it can interact to shift the biogeochemistry of these systems. So this is really important in terms of inorganic as well as organic materials. And all of these drivers of change have ways of antagonistically or synergistically influencing these materials. So it's really difficult to quantify these effects, these positive and negative effects, over very, very large scales. And I just told you that the permafrost underlies 25% of the northern hemisphere, and most of these places are relatively inaccessible. So to pull back for the second, in the Arctic specifically, there's these competing perspectives on the direction of change. And this is primarily a result of the way that we study Arctic systems. So at the plot level, Arctic ecosystems are seen as relatively ecologically closed, where they don't want to give up a lot of the nutrients that they've worked really hard to attain. So less than 1% of any photosynthetically fixed carbon is lost from a tussock tundra. And isotopically labeled nitrogen studies show that these systems work really, really hard at the plot level to hang on to that nitrogen. So, but if we think about big regional scale studies, things like remote sensing or these studies that happen over a very, very large scale, Arctic systems are actually seen as relatively open or leaky. So that means that there's these really disparate predictions of what direction the Arctic will shift in as a result of the way that we study it. So the work that I do is really trying to bridge these scales by using the watershed unit as a way to sort of integrate large plot level and regional scale understanding. So we did this meta-analysis just to give a kind of example from the Kuparuk River basin where I work. And we know that the subcatchment level is actually very, very underrepresented in Arctic work, where there's kind of this bias towards plot level studies, like at these very, very tiny scales or very, very large scales. So from my perspective, right, I like to ask, like, what is the river actually telling me? So I want to invite you to look at this system where the flow is moving towards you and ask, what is the river telling you as it's flowing past you? And what you would say probably depends on your unique perspective, right? So if you were a hydrologist, you would say the water is telling you where it's been from both the surface flow paths as well as the unseen flow paths under the surface. If you were a biogeochemist, you would say it's also the water is carrying with it a lot of material that it's picked up during its journey to you. And if you're an ecologist, right, you might think, okay, that material has been transformed as it's moved towards you through microbially mediated processes, or even in the Arctic, a lot of sunlight winds up being a driver of these reactions. So ultimately, we can use river water to tell us about these issues of scale and what's happening on the landscape based on what the water is carrying with it. So I use a lot of this basic theory of using streams and rivers as these spatiotemporal sensors to sort of bridge these scales and give us a better understanding of what's happening on the landscape as a result of climate change. So for the remainder of my talk, I'm going to talk about two, again, NSF-funded work projects. The first being my postdoctoral fellowship, and the second being a bigger collaborative project that I'm a co-PI on. So first, we'll talk about how, when, and why Arctic systems might be more leaky to carbon and nutrients using storms. So it probably doesn't surprise anyone in this audience that storm events are these hot moments of material export. They have a lot of potential for water to just flush the system. So I don't know how many people has tried storm chasing, but it's really, really challenging to actually do in a way that is meaningful. So in the Arctic, it's even more sketchy because it's just really dangerous, like to actually go and get samples during these periods of time. So usually, these storm chasing is really, really challenging. So as a solution to this, we deploy high frequency sensors to actually capture what the river is carrying with it every 15 minutes. So we use S-scan spectralizers to tell us about the total dissolved organic carbon, dissolved organic carbon, as well as nitrogen species such as TDN or total dissolved nitrogen, as well as nitrate. We co-locate this with a discharge monitoring as well, so that we can actually get fluxes of these materials and not just concentration. But what we end up with is this amazing time series across the entire season of data every 10 to 15 minutes. So these data can tell us a lot about what's happening across the season over years. This contributes to long-term monitoring, but as from my unique perspective, I'm really interested in how these signals can tell us a lot about the material sourcing on the landscape. So to give a little bit of, to step back a bit, we deploy these in watersheds that are associated with Toolik Field Station, which is on the north slope of Alaska. So I do just want to mention that the Toolik Field Station is on the lands of indigenous, or the homelands of indigenous peoples, and I just want to recognize that for a moment. We deploy these sensors in 3 core watersheds that are actively monitored by the Long-Term Ecological Research Station at Toolik. The first being the Kuparuk River. So the Kuparuk is one of the best studied Arctic rivers in the world. It's very low gradient. It has really high biomass. You can see how green it is. Really low N. It's very N limited, and it's very abundant in carbon. We also deploy this in Oksrukuyik Creek, which is similarly low gradient, low N, high carbon content. But what's really unique about this is the idea of the stream-lake interactions. So I'll show a map in a bit, but essentially within the watershed network, there's a bunch of lakes that are nested within the watershed that are connected to the streams and rivers in that network. And then we also deploy this in Trevor Creek, which is sort of our outlier in some ways, where it's our alpine watershed. It's very, very high in nitrogen, given nitrogen deposition, or like relatively speaking for the Arctic, and very low in carbon, because there's not a whole lot of organic matter accumulation in the hill slope. So these colors, blue, orange, and green, will be consistent throughout the remainder of my talk, and I'll try to remind you of those moving forward. So when you go to the doctor, you would get an integrated measure of your health, right? If the doctor takes your pulse or your blood pressure. And we can use these high frequency data to tell the same thing about the watershed. So essentially, with these high frequency data, we can use concentration discharge relationships to tell us a little bit more about material sourcing on the landscape. So we do this to essentially explore the watershed's pulse. So we have a hydrograph, right, from the discharge records. We also have a chemograph from the high frequency sensors. So what we can do is we can then convert that into a concentration discharge relationship. So when you take the log or put this relationship in log-log space, you can then calculate the slope of that relationship as the exponent of a power law. Essentially what this tells you is the material source pathways as water and material has moved towards the watershed outlet. So this relationship is really nice because it tells us a lot about what's happening to move material towards you. So this value of beta can tell us a couple of different things. You can also do a lot of work with the relationship and the shape and direction of this, but for simplicity, we'll just focus on the linear slope. So if the beta is positive, meaning that concentration is increasing as discharge is increasing, this indicates an enrichment response, where essentially material is sitting on the landscape that just needs to be hydrologically activated before it moves to you. So it's transport limited, it enriches. When concentration increases proportionally to discharge, this doesn't just means that there's not a hydrologic effect, so that it's considered chemostatic and the beta is close to 0. When concentration decreases as concentration increases, this indicates a dilution effect, meaning that the source or material pathways are such that material is actually diluted before it reaches the outlet. So this results in a negative beta. Okay, so as an example of how this might look, this is data from the Kuparuk River for a week. So essentially what we do is we splice all the time records by storm events. So I do this using a filter approach, which I'm happy to talk about afterwards, but I won't go into the details about it today. But we do this to delineate all of the storm events and do this concentration relationship at the event scale. This is what this ends up looking like. And obviously, so this is for DOC. DOC is enriching, that beta is very, very positive. So we repeat this systematically for every storm event, for every solute, for every watershed ad nauseum. So we get this really nice data set. And as a stream ecologist, I was really interested in whether or not we would see the seasonal effect of the active layer thaw as the season is progressing in that CQ relationship. So what I mean by this is that the active layer in the Arctic at the early part of the season is very, very constrained as it's mostly frozen. So water movement is constrained to that top layer, essentially through these highly organic layers and an old plant material as it's before everything greens back up. As the season progresses and the active layer thickens, it thickens through a mineral horizon that is no longer like highly organic. And so we figure that this seasonal shift as the active layer is thickening, that this would change the CQ relationship such that DOC would be widely available and heavily enriching at the beginning part of the season, and then became more chemostatic towards the end of the season as water is mostly moving through these mineral horizons. For nitrate, we sort of expected that this would be related to plant productivity that would peak kind of in the middle of the season, meaning that there would not be a whole lot of nitrate to move through the systems, so that storms in the middle of the season would basically be the most diluting. Okay, so just to prime you on how to read these, this is essentially plots of CQ slope over Julian day, where each point will be a single storm event for DOC as well as nitrate. So anything above this line, the 0 line, indicates enrichment. Anything below indicates a dilution effect. So in general, we kind of thought that the seasonal effect of the active layer constraining water flow paths would be really evident in these CQ responses. But I mean, no matter how you look at this, like maybe if you blur your eyes a little bit, you would see a seasonal response. But we were actually really surprised that this was not evident whatsoever. I mean, DOC was consistently enriching across our Kuparuk watershed, and nitrate was consistently diluting. And this was also consistent in the Oksrukuyik watershed, where we see like this very consistent enrichment of DOC and dilution of nitrate. So for ease of interpretation, instead of thinking about the scatter plot of things over time, here we're just looking at the annual box plots of the CQ, the mean CQ relationships across all of the storm events. So what's really clear is that there's actually this landscape driver of CQ responses, where the tundra and lake watersheds through the Kuparuk and Oksrukuyik consistently enrich DOC, while the alpine watershed does the opposite. It actually dilutes that carbon because it's not available on that watershed. And for nitrate, we see this dilution effect of the two watersheds, Kuparuk and the lake dominated watershed, and the alpine is consistently enriching in that nitrate. So at the watershed scale, we don't really observe this clear influence of the seasonal active layer thickening. So this was actually very surprising in many ways, until we had a really wonderful graduate student join the team who thinks a lot about how water is sourced from multiple sources on the landscape. So this finding is actually very consistent with the idea of variable source areas, the fact that water and where it's coming from is dynamic across the course of the season. We weren't necessarily expecting that for an Arctic system, which we viewed as probably more simplistic than we probably should have. But essentially that these change in flow paths and water sourcing, because they're dynamic across the season, sort of blurs that effect, at least at the CQ event scale. So Amelia Gross is the PhD student at MSU who's working on this, essentially trying to parse out the different sources of water on the flow paths of water using hydrologic tracers as part of her PhD work. So again, this is like part of the best part of my job now, is getting to work with really awesome students who use tools that I am still, getting to learn. Okay. So the important thing that came of this work is that there's this consistent pattern across the Kuparuk and Oksrukuyik, and then a distinct pattern of Trevor Creek, the alpine site. Just for ease of interpretation, I'm going to move this into quadrants. So this kind of gets us back to where our system's more leaky to these materials. So in this quadrant, this allows us to compare nitrate and carbon export, where the location in these quadrants essentially says, are these being exported synchronously or asynchronously? And what does that mean for carbon and nitrogen stoichiometry? So every point that I show on this plot will be a single storm event. So when we consider these constituents together, what's really clear is that there's sort of this gradient of responses when it comes to carbon and nitrogen. And that's really exciting in some ways because it allows us, again, to compare these landscapes a little bit more aggressively. So Trevor Creek always winds up in this quadrant where nitrate and DOC are asynchronously exported. But this Trevor Creek system is very, very leaky to that nitrogen. So it's not necessarily able to hold onto the nitrogen, whereas the Kuparuk storms are flushing a lot of carbon, regardless of when those occur. So the Kuparuk is exceptionally leaky to that carbon and tends to hold on to nitrate. You'll notice that Oksrukuyik, while it is sort of in this lower quadrant, it does tend to sort of act in between the Trevor Creek and Kuparuk sites. And we think that this is heavily related to the fact that Oksrukuyik has this lake influence on the watershed, which I'll get at in a minute. But essentially, this is this not necessarily intuitive relationship where you're actually taking your CQ measurement might really change the stoichiometry of your system based on when you might get a storm event coming through. So I want to pause now and actually reflect on the influence of an REU student on this project. So this is Abigail Wreck. So she was an REU student at the Toolik Field Station in summer of 2019. And she was really interested in how lakes might change material availability to stream biota. So she, in addition to working in the Oksrukuyik watershed, she sort of focused a lot on the I-series watershed, which is within walking distance of Toolik. So to give an aerial view of this, the I-series watershed has this really nice system where we have a very distinct inlet, a lake, and then an outlet. And again, there's Toolik. So it's really nice, it's walking distance. But she essentially did these really nice nutrient assays where she put nutrient diffusing substrata out at the inlet and the outlet of this I-series lake to understand how does the presence of the lake potentially influence nutrient availability. What's really amazing is that the inlet and outlet biofilms, I won't go into the details of the NDS too much today, but the inlet and outlet were very distinct in terms of their ability to respond to a nutrient amendment. So this suggests sort of this variable response that the community or something is different between the inlet and the outlet. In addition to the NDS, Abby also helped us place a scan at these same locations. And one thing that I want you to remember is that the Kuparuk was in this quadrant, right, in terms of the CQ responses. And this is exactly what we see with the inlet response, which is essentially just behaving like we would expect a tundra watershed to behave. But the outlet of the watershed is behaving completely different, where it's actually enriching in that nitrate response and diluting carbon. So this sort of helps us explain why the Oksrukuyik sort of fell like right in here, kind of in between these inlet and outlet responses, because water is continuously moving into the lake and then out, and the lake is essentially behaving as a reactor or a buffer to those signals. So this was really exciting in a lot of ways. I just want to highlight that this was primarily work of an REU student who was really inspired just by looking at the landscape. This ended up seeding a master's thesis, it seeded a proposal, and we're continuing a lot of this monitoring of the lake inlets and outlets at high frequency, primarily just because we had the opportunity to work with Abby. So I am an ecologist, but I do think about hydrology. And one thing that my postdoctoral advisor, Jay, always says is that hydrology or physics gets the work and then biology gets it done. Meaning that, you know, hydrology might be a really strong driver of these responses. So one thing that he was really interested in that, you know, we tried to tackle is parsing the effect of hydrology, moving material versus the source of that material being available for that hydrologic transport. So essentially what we tried to do is think about the CQ response parsed by the discharge quantiles. And what I mean by that is at low flows or at base flows, we would expect that most of the water is coming from these unseen flow paths through potentially organic rich systems. And so we would expect, you know, at base flows that we would see very, very high enrichment, given that these flow paths are coming from these organic rich layers, and potentially very, very high dilution of nitrate, given high plant uptake, because the system can take that on. At intermediate flows, or like once a storm hits, we might see more surface flow paths and runoff generation that bypass the reactivity of the hill slope or the land, such that we would actually start to see more of a chemostatic relationship with carbon as that system is starting to dilute, and then more dilution of that nitrate, just given that these plants are very nitrogen hungry and they're starting to take on that excess nitrogen. Then at extreme high flows, you know, we would see a lot more surface runoff and dilution of carbon and yeah, continuing dilution of nitrate. So we did this by parsing our CQ responses by the discharge quantiles by year. So these plots essentially show that CQ slope across those discharge quantiles annually. Which we did this annually because the discharge changes quite a bit year to year. So for the Kuparuk, while we might have expected, you know, this still approaching chemostasis for carbon, what we saw is that more water just means more carbon moving to the system. And that's actually really exciting in some ways, because while it's the opposite of what we might have expected, it's kind of this intuitive thing that as long as there's water moving through the system, there's carbon flushing into the stream channel. And we see the opposite for nitrate. So in general, across most years, we see this very, very strong influence of hydrology driving material mobilization into the stream channel. So this is sort of what we expected, but for Oksrukuyik, again, this lake-dominated system, we don't see any influence of hydrology on the CQ response for nitrate or for DOC, pretty much across any years. So while this might not be super intuitive, we think that this, again, has a lot to do with sort of these stream lake dynamics. As water is moving into a lake, into a different reactive zone and moving out, then it has this sort of buffering effect on the CQ slope that it doesn't matter how much water is moving through the system, there's some storage capacity of these lakes on the landscape that essentially bypasses what we might expect for the Kuparuk. Okay. So to wrap up this section, I told you a little bit about how storm events are sort of these hot moments of stream export for both carbon and nutrients. So storm events are these really important moments to capture. And then I talked about how landscape templates, so the Kuparuk is very, very leaky to carbon, whereas alpine systems tend to be very, very leaky to nitrate. Whereas the lakes on the watershed have this capacity to sort of buffer or filter out these signals that we're still working on exploring. And then that event size can also control the ability of the system to actually mobilize that, at least in our tundra watershed, and it doesn't necessarily apply. That theory essentially falls apart when there are lakes on the watershed network. But one thing I want to mention before I move on to the other section is that when we're talking about in the season matters a lot. And what I mean by that is this is a typical Arctic field season. So we did this meta-analysis of Arctic systems, Arctic field studies in the Kuparuk River Basin. And essentially there's this really well-defined season where most Arctic researchers flock up to Toolik. This is primarily a result of the semester schedule, and also it's really expensive to head up there. It's like 120 bucks a day per person to be there, plus the helicopter hours. So it adds up. So most of the time we're studying the Arctic during this peak of the field season. But when we're talking about a changing Arctic, this is sort of when we think about a typical discharge regime in the Arctic. This is predicted to move far earlier, and we're going to experience more storms later in the season when we're not doing a good job of capturing what's happening on the system. So I think understanding the shoulder seasons, both with these CQ responses as well as just a better planned monitoring, will be needed as we move things forward in the Arctic. Essentially, how can we start to capture between this frozen state and when things are flowing? And I think that high frequency sensors are one way to do that. It's not easy necessarily, but it is one part of the solution. Okay, so I'm going to jump now to part 2 of my talk. So this is a very large, different type of project, which is highly collaborative, led out of BYU, but has collaborations at MSU, University of Vermont, as well as the National Parks of Alaska. So the project is lovingly titled the Arctic RIOS project, which stands for Arctic Arctic River Integrated Observations through Synoptic Sampling. So it's this big mouthful, so we use this acronym. So if I use RIOS throughout, that's what I'm talking about. So the core of the RIOS project is to constrain what is happening to carbon and nutrients that are being released from these across the permafrost region. Now, if I revisit this figure that I showed you earlier, what's really amazing is that the outlet will tell you, what's happening on the integrated watershed of the entire system as it mixes and moves to you. But the watershed is essentially this patchwork of material sources and sinks and processes that are changing what material actually makes it to the outlet, right? So we can essentially use the spatial distribution of these patchworks to tell us a little bit more in detail about the patches that result in the chemistry that made it to certain parts of the channel. So essentially, we do these synoptic sampling campaigns in addition to the watershed monitoring outlet to give us a bit more detail about what's actually happening distributed throughout the entire watershed. So we capture these, again, using synoptic sampling approaches. And what that means is we get in a helicopter, which is really fun and exciting and scary, but we go out and in a span of about 5 to 6 hours, we capture about between 40 and 60 points in a single watershed in a few hours. So we capture pretty much everything. If we can measure it, we do. We have a whole periodic table of elements that we end up measuring, but I'm just going to focus on carbon and nitrogen for the majority of today, just in the interest of time. This data is public, so if you are interested in exploring this, it is public and available on Earth System Sciences data, as well as the Arctic data portal. So please use it. We can ask these really amazing questions about what is controlling carbon and nitrogen in these systems. How representative is one subcatchment relative to another, and how do they contribute to what we see at the watershed outlet? We can also understand from the opposite end of if we are not able to actually go out and measure these things, which takes a lot of time and effort, can we use remote sense products to actually tell us a little bit more? How well can we predict stream chemistry from that? And we can also do some really amazing things about recovery from major disturbance. So we, again, conduct this via helicopter. So this has a lot of different advantages. It means that we can access these field sites that are really otherwise inaccessible. They're not hikeable. But this also gives us this really nice bird's eye view where we get to know our watersheds really, really well. The other advantage is that we have a lot of statistical power. I just told you that we get 120 subcatchments, we get all of these different constituents across 3 watersheds across years, and we do this multiple times a season. So we have a lot of ability to tell about what's changing, why things are changing, and we also have these pictures, right? We have this view of what might be happening on the landscape to result in these changes in stream chemistry. So one thing we can do with these data sets, again, is try to understand how representative is one stream relative to another. So using these data, I'm really interested in trying to understand what contributes to the watershed budget. What contributes to how much carbon is actually exported or how much nitrogen. So are the things that are influencing stream chemistry diffuse, meaning are these changes or are these influences happening everywhere? Or is the stream chemistry driven by a few outlier points on the watershed? So this is scaled DOC concentration as contributing areas increasing. So essentially these are small subcatchments that are then mixed as they make the water makes its way to the catchment outlet. So I'm not showing units here because they are scaled just so that all of the watersheds could be on the same plot. But here we can start to ask questions about representativeness, right? So we might expect that we would see really, really high variance in these small contributing area subcatchments because they're highly connected to the landscape. So you have really highly variable smaller subcatchments that are draining really, really different little tributaries. And then as things mix, they make its way to the outlet. But one thing I want to highlight here is this idea of representativeness. So this is a Naviat Creek that compared to systems that are of similar size, it tends to be this outlier for pretty much every constituent, especially DOC. So it's always exporting a little bit more than we might have anticipated. So I just want to mention that data from this subcatchment it's a tributary of the Kuparuk, is inputted into regional and global climate models that predict the direction of change in the Arctic. And yet it is this sort of non-representative subcatchment compared to systems of similar size. So that's problematic, right? Because we might be either over or underestimating these exports based on this one non-representative system. So what more can we do with these data sets? So we use essentially this tool that helps us understand the contributions of a single subcatchment to the watershed mass balance. So we use this wonderful metric called subcatchment leverage, which is essentially analogous to statistical leverage, where you might have one or 2 points that sort of are biasing your regression analysis. This is a technique that's borrowed from landscape ecology, where we're applying this to each subcatchment that we capture the stream chemistry for. We do assume a specific Q that's equal throughout our subcatchment. And essentially what we get is a leverage in percent. So how much relatively is this 1 point contributing to the distributed mass balance of your system? So the way to read this is that anything above 0 means that that subcatchment, relatively speaking, is a net source of that material, that it's exporting more than we might expect. If something is along this 0 line, that means that things are relatively conservative, meaning that everything is pretty well distributed, well mixed. There's not one or 2 subcatchments that are really contributing to that distributed mass balance. And then if it's lower than that 0 line, that indicates a net sink, that it has lower concentration than you might expect of a similarly sized watershed. So this is looking at leverage for DOC from a sampling that we did in 2017. So what you start to notice is that every point across contributing area is pretty much along that 0 line, indicating that DOC is pretty much leaky everywhere. There's not one or more points that are really driving that relationship. So we can visualize this spatially. So this is the map of the Kuparuk River Basin. Flow is moving towards the upper left. And everything is pretty much this yellow color. So anything that would be blue indicates a sink. Anything that's red indicates a source. So this is, again, leverage in percent. If we repeat this, which we do, in August of 2017, we start to notice a couple of points that are a strong source of DOC. Again, indicated here on the main stem of the Kuparuk. So again, the benefit of doing these synoptic sampling campaigns is that we get to know our watersheds really well. And we know from visual, being in the air, we know that a thermokarst is starting to develop on this portion of the watershed that pretty much opens up almost annually, resulting in these increased carbon concentrations. So the ability to have eyes on the watershed is this really important added benefit of our synoptic sampling campaigns. Okay, so to fly over the ridge, this is data from the Oksrukuyik Creek watershed, again, our lake influence watershed, again, from the early portion of the season in June. We don't really see any, oh, now it's working. Okay, now my clicker is working. Okay, so if we map this, again, everything is sort of this yellow color. Here you can kind of see the influence of those lakes, and flow is moving this way towards the upper right. Oh gosh, I can use this now. Okay, so if we resample, we start to see these strong sources of DOC that are a little bit more distributed. So what we have found is that a lot of these source areas are just downstream of those highly lake-dominated subcatchments. So this essentially means that these lakes are sort of acting as this reshuffler of our system. To give you a better visual, right, of what this looks like this is a picture of Oksrukuyik watershed in sort of this upper portion right up here. So essentially, we can use these really nice statistical tools to tell us a little bit more than subcatchment leverage. So we use a Spearman's rank correlation to tell us a little bit more about our ability to predict solute. If we take a sample at the early season, can we predict that same concentration at the later portion of the season? This we've termed spatial stability or instability. And for the Kuparuk in blue, and Trevor Creek in this green, these signals are relatively spatially stable. So we have a pretty good idea of what the concentration at a given subcatchment will be based on the early season sampling. In Ox, it's more of a shot in the dark. There's a lot of spatial reshuffling, and this is really consistent across our years and across our seasons, where the lake's watershed tends to have this instability in its water chemistry. So we have further evidence of this because we have so much data. So this is a better idea of the Oksrukuyik Creek watershed right here, and you can see how lakey it is. In the recent years, we've done a lot more than we initially did with capturing the lake inlets and outlet chemistry. So in our repeated synoptic sampling, we've collected DOC and nitrate from a lot of different lakes, and we've specifically focused in the last year or two on capturing the inlets and the outlets. And then one thing, these are arranged by lake size, by the way. I know this is a lot of data that I'm showing you, but what I want you to notice here is that there are some lake systems where DOC is produced and nitrate is removed, at least in terms of just in situ concentration. And then there are other systems, they tend to be larger, where DOC is removed and nitrate is produced. So the patterns of the inlet and outlet are not always consistent. We think this has a lot to do with lake residence time and how much time water and material has to be processed. But essentially, lakes can act as both an amplifier of these signals as well as a filter. So they're not always consistent in space and time. We think this has a lot to do with the instability that we see, at least at the watershed scale. Okay, another way that we've been using these data is to think about the opposite question of how well can we use remote sensed products to essentially predict stream water chemistry in a changing Arctic. These systems are really remote, so we essentially, these data are really unique that we're collecting, but it's not practical to do this across the entire watershed or the entire Arctic. So this is Tanner Williamson. He's a postdoc on the project. He became really interested on the influence of land cover and season on these signals. Oh my gosh, okay, I need to not press this because I will turn off my screen. So I'm gonna skip through this, but Tanner used land cover data that we have from GIS specialists at Toolik to essentially ask how well can we predict our whole periodic table of elements. He put this into a boosted regression tree, which is a machine learning technique to essentially try to understand the relative influence of season, land cover, all of these things on stream water chemistry. So the BRT approach gives essentially relative influence of all of these different things. I know this is, again, very, very overwhelming. But what I want you to notice is that the seasonal or the influence of these parameters changes based on the solute of interest. So slope tends to be the strongest predictor for carbon, whereas for nitrate, the most important parameter or the most influential parameter is season. In addition, we also get our ability to predict those solutes with good accuracy changes by solute. So our ability to predict based on this BRT model carbon concentration is really pretty good. In terms of an R-squared, it's over 0.9. However, for other things like particulate phosphorus, our ability to predict that based on the landscape data that we have is actually pretty bad. So there are some solutes that we do actually need to go out and measure and do a better job of measuring, while there are some solutes that we could actually just use, at least in the Toolik area, we can use some of the landscape data to help fill in some of the gaps. Okay, so I know I'm running a little behind on time, but beyond characterizing these patterns across season for the Toolik area, we have also been really interested in trying to understand response to disturbance. So I'll tell like a very, very short story about that. This is a really challenging thing to document, right? Because you actually have to kind of be there to understand the disturbance, especially in terms of wildfire. So this is the Toolik River fire, which happened in the mid 2000s, and it burned this massive area of land. It was one of the biggest tundra fires and has been pretty extensively studied since. However, this is work that was spearheaded by Ben Abbott of thinking about what happens 10 years later. Can we still detect the influence of that burn? So we sampled these subcatchments that had been unburned as our kind of control because we didn't have the measurements before, as well as sort of severely and moderately burned subcatchments. So the result of the wildfire, at least at the Toolik fire, is that there's a consistent active layer thickening that happens post-fire. So essentially, once a fire happens, you have this very, very thick active layer that essentially degraded some of that permafrost layer. So this area, again, is very, very remote. So we used the unburned catchments as a control and the severely burned catchments as our treatment here. But this is very far from Toolik and inaccessible otherwise besides helicopters. So to actually sample these, you can't land. Like normally during our synoptics, we land, we get out, we take our stuff, we take a sample. But here, we couldn't waste fuel. So instead, someone, normally not me, because I don't love doing this, is you take the door off the helicopter and you stick a modified tent pole and you jab the water with that has a bottle on it, and you bring that up, and then you go to the next site. You don't land. So that's been named the dongle. If you want to read more about it, it's actually published. Okay, so in general, by comparing these unburned and burned catchments, what's been really amazing is that even a decade later after the fire, we still see this persistent material loss from these systems. So this is just carbon, but this is consistent for other constituents as well as in terms of nitrogen, phosphorus, organic, inorganic forms. We see this persistent leakiness of the system. And this is despite some recovery of the biomass, at least below ground and above ground. So we do think that the fire, by changing the active layer depth, it's fundamentally shifted where water can flow in these systems, where it essentially it bypasses the layers at which it could be taken up, like nitrogen and carbon. And so Chi-Wen is a graduate student at BYU, and she's been working a lot on trying to parse this apart, again, using hydrologic tracers. I don't have a good picture of her, so I didn't include a picture on the side, but she's been really wonderful to work with on this. Okay, so to wrap up this section, I talked briefly about how we can start to use these really unique data to tell us a little bit more about the representativeness of the spatial contributions, whether or not these effects are discrete or diffuse. We also talked a little bit briefly about how we can start to use remote sensing products to try to understand stream water chemistry. So coming at it from the opposite, like where might you not have data, and can you predict stream water chemistry from parameters that you might get from Google Earth? And then that spatial synoptic sampling can start to help us understand the long-term consequences or the legacies of disturbance. So and I do want to mention, so a lot of these data are public and available on the Arctic Data Archive. So if you're interested in them, please find them. Otherwise, we do have at least one, maybe two more field seasons to go on this project. And so there's a lot of products that are forthcoming. So keep in mind if you're looking for data. Okay, So Arctic regions make up some of the last remaining wilderness in the world. And hopefully what I've told you today is that, and I've convinced you of, is that flowing water in these Arctic systems does tell a story of how systems are changing. So they make up some of the last remaining wilderness on Earth. They're this amazing place to travel and do work, especially during the summer. I've never been in the winter. They're this home to these amazing plants that are cold tolerant, and obviously these amazing, megafauna and indigenous peoples. But this goes beyond the Arctic. That we're experiencing, a lot of significant ecosystem change from changing hydrologic regimes as a result of climate change, altered land use and land cover, which shifts aquatic terrestrial linkages and essentially fundamentally shifts biogeochemical fluxes. There's a significant loss of biodiversity in terms of food web degradation and novel species assemblages as a result of invasive species, as well as the influence of novel inputs that fundamentally change ecosystem structure and function. So hopefully one thing that I've told you today is that rivers can help us understand and document how systems are changing. And I think it's really important that we try to understand and listen beyond just the Arctic, which might seem very far away. We can use the streams in our backyard to tell us a lot about how the systems are shifting. Okay, so with that, I just want to wrap up and thank my collaborators, and I will happily take any questions. And sorry, I went a little bit over time.
I want to just remind people that are online you can put your questions in the Q&A. And I think I'll start. I think we have a Q&A here, a question here. Let me see. Someone is asking, do you have any data on the age or other characteristics of the DOC to explain some of the dynamics you observed?
Oh, that's a really good
question.
That
was from Peter Groffman.
Peter Groffman. Yeah, hi, Peter. Good to see you virtually. So yes, we are taking carbon quality data, so as well as quantity. Those data are being worked up by a graduate student at BYU. So we have data from an aqualogs, including EAMS, as well as other, quality from a spec. And we also are planning on taking C14 to understand the age of the carbon. We did not do that last year as that was, we were initiating the funded portion of the project. We do have funding to do that, and we have plans to do that this upcoming season. So that is something we have in the works and plan to add to the field seasons that we have left on the project.
Questions from the room, and just remember to repeat them.
Okay.
Thanks. Go ahead, Gary.
Remarking on the graph of the watershed size versus GSE concentration, they were really variable in small watersheds and then sort of averaged out in larger watersheds. We've seen that same pattern in nitrate in temperate zone watersheds. And I was wondering, how do you distinguish between just an averaging effect of all those new watersheds going into the larger river versus some sort of in-stream processing going on in the river that sort of moderates them?
Yeah, that's a really good question. So a lot of that, we sort of use the subcatchment as our unit, and then we try to attribute that with what we see on the watershed. But there is sort of this disconnect between that and the function of, is it because there's a high nitrate uptake in this, this one portion of the reach that results in really low nitrate concentrations. So we are, this is a work that actually Abigail Wreck is leading. She's working on some uptake experiments distributed to try to understand how much of it this is resulting from in-stream processing versus, what's happening on the landscape to input that into the system. And paired with some of the tracer tests that Amelia Gross is leading, I think we'll be able to understand how much of this is a landscape effect versus how much of this is in-stream channel processing. So I hate to leave you on a cliffhanger, but that's something we've been thinking a lot about, and how do we parse that apart? And yeah, we're using a combination of methods to do that. Steve.
Talk about wetlands too much, except for that land cover analysis by Williamson, but that was like % wetlands and all the catchment.
Yeah.
And in other watersheds, wetlands are frequently disproportionately important, as you probably know. You know, a source of DOC and a sink for nitrate, for example. If you knew how much the water went in and out of wetlands, the spatial distribution of that, do you think that would be enlightening?
Can you repeat the question?
The question, yeah, sorry. The question was, if we knew the inputs and outputs of wetlands, would we be able to tell us? Can you repeat the question? Sorry, I'm like doing a bad job repeating the question.
So might that help you explain spatial patterns across watersheds, differences among watersheds, if you knew how much water is going through wetlands?
Okay, yeah, so if we knew wetlands, would we be able to understand the spatial patterns? So in these systems, wetlands are kind of a loose term. It's not like a wetland, like an isolated wetland or even a riparian wetland that we would see here in the temperate zone. So I have a feeling that it's probably less important here to understand wetlands than it is to understand sort of the way that water is routed. So sort of what Amelia is doing with trying to understand, is water constrained to the surface, or is it coming from these like deeper, longer flow paths? So maybe on a couple of the subcatchments that have high wetlands, then it would be really important, but I would think distributed across these systems, it wouldn't be as important as you might expect here.
Other questions? I have a question.
Yeah,
That's okay.
for it.
So the at the end you talked about the fire and the influence of fire on the export of nitrate, DOC, and everything. And that was like 10 years, and you said that there's still a legacy effect of the fire. And I'm just wondering if you all have talked about like how long, is it an irreversible change, or is, because we're seeing increased fire in the Arctic, is it something that is gonna, like, does it take 20 years for that to go back, or is it once it burns, it's in a new state?
New state. Okay, so I guess they heard the question. Okay, so I think in this system, it's probably semi-unique because the Anaktuvik fire was a very big, very wide-ranging burn. In terms of the length of time, we haven't really thought about that in terms of how long it would take the system to recover, primarily because these data sets that at least compare before and after are so limiting in these systems. I have a feeling that this probably takes a significant amount of time to recover in terms of to prior export regimes. But yeah, that's a really good question. And those data, they were not part of a funded project. That was based on an opportunistic LTER gift of helicopter hours. So, being able to actually document that is not something we've been able to do quite yet. And I think that's a really good question of how long it would take. So yeah, my answer is I don't actually know. I don't think anybody really knows.
Good long-term ecological research.
Yeah, great for a long-term.
Just as we have another moment or two, I'll ask a question that Kathy Weathers asked online. Do you have any results from DON? How does it behave differently or similarly to nitrate? And of the total nitrogen that moves through the system, what proportion is nitrate versus DON? Oh,
that's a really good question. Yeah, so DON is the dominant form of nitrogen in this system. So it's mostly DON that's part of the DOM pool. So when we do the CQ dynamics of TDN, we can also do this for DON and get good time series of DON. And it pretty much matches the DOC patterns, that it's always kind of flushing the system. So I think DON is sort of this unrecognized source. I think some people think of it as relatively boring in the system, but I think it's the dominant form of N. At least in the Kuparuk and Oksrukuyik, it's the opposite. Nitrate is the dominant form in Trevor Creek. So I think in general, DON is sort of this untapped pool of nitrogen. And I think there's a lot that could be said about photodegradation of that DON to inorganic forms. So I didn't mention this throughout my talk, but the influence of sunlight, like these systems are 24-hour daylight at the peak of the season. And so I think photo processing, like photoammonification is a real source of inorganic nitrogen to the system that is sort of an untapped topic. So, but yeah, DON is really important.
Great, thanks so much, Ariel. Yeah. And thanks again for coming and giving a great seminar. And Ariel is around if you want to meet with her. She's having lunch, and we're having a gathering later today. So please join us. And thank you all so much for coming. And thanks, Ariel, for a great seminar.
Yeah, thank you.